Good morning, good afternoon, good evening, and for some of you, even good night, everyone. And thank you so much for tuning in from all those different places to our webinar series, New Tools of the Trade in the Life Sciences, Technologies that Enables Breakthroughs. And today we will hear about developing glycoproteomics and glycoengineering approaches to investigate protein glycosylation. Okay, my name is Renate Weizbauer. I'm one of the leads of this webinar program here at Bioprotocol, which is an open access online journal with the mission to make research more transparent and reproducible. And before we begin this webinar, let me share a couple of quick housekeeping announcements. If you have a question, you can post them in the Q&A or in the chat section throughout the talk, and Lorenzo will address them at the end. And this webinar will also be recorded and posted on our website later. So if you want to revisit it, you can do so later. All right. It is now my very distinct pleasure to introduce the moderator of today's webinar, Katarina Madunic. I hope I got that one. Good. Who is a postdoctoral researcher with profound interest and passion for glycobiology and glycoanalytics. She obtained her PhD from the Leiden University Medical Center in the Netherlands, where she worked on methods development and discovery of novel colorectal cancer-associated glycan signatures using mass spectrometry. She's currently working as a postdoctoral researcher at the Copenhagen Center for Glycomics in Denmark, where she focuses on understanding cellular processes related to the function of cadherin-specific O-mannosylation in cancer. And with that, Katarina, thank you so much for being here today, and the floor is all yours.
Thank you so much, Renee, for the introduction. Now it's my turn to introduce our today's session. And first, I'm going to tell you a little bit about glycobiology. Now there's growing evidence that the glycans on proteins regulate diverse protein interactions and biological functions, but they're so complex that technical barriers so far have left the glycomics field underexplored, but with a huge potential for discovery and biomedical applications. And here at Copenhagen Center for Glycomics, where both the speaker and me are working, we are developing novel technologies to overcome these barriers. And we use genetic approach to glycomics in that sense. We use gene editing for glyco-genes to dissect enzymatic pathways and characterize them in different disease variants in cell-based models. We also use advanced mass spectrometry to study glycan structures and map their site-specific locations on proteins. And the aim is to apply this expertise to investigate the functional roles of glycosylation and identify diseases associated with defects in glycosylation. And now it's my pleasure to introduce our very, very talented postdoctoral researcher, Lorenzo Povolo, and he will give you some further insights into this subject. Lorenzo obtained his bachelor degree in biotechnology from University of Trento, Italy, and a master degree in molecular biology and genetics from the University of Copenhagen. And already during his master's, he got familiar with mass spectrometry, so he joined Copenhagen Center for Glycomics in the group of Adnan Halim. Here, the focus of his PhD studies has been on investigating the biosynthesis of protein O-mannosylation in humans. He contributed to the discovery of TMEM260, a new independent pathway for glycosylation of specific receptors. In his PhD studies, he used genetic engineering tools to dissect initiation of O-man glycosylation, developing sensitive glycoproteomics tools. He recently obtained his PhD, and now he's a postdoc focusing on characterization of O-man pathways using structural biology tools. Lorenzo, the floor is yours.
Thank you very much, Katarina, for the very nice introduction. Also, thank you, Renee, for organizing this webinar. It's my pleasure to be here tonight, to give you this webinar. And while I'm sharing my screen, I would like to thank everyone that is tuning in for this occasion. So I will get started. The pointer. There we are. So I hope you can see everything. So I would like to welcome you formally to this webinar on developing glycoproteomics and glycoengineering approaches to investigate protein glycosylation. So I would like to start by giving an outline of today's presentation, just to give you an idea of what is going to happen in the next more or less 45 minutes. So I would like to start by giving an introduction to the field of glycosylation and protein glycosylation. So just to give an idea of what we are actually studying with the glycoproteomics and glycoengineering tools. So this will lead me into an introduction to mass spectrometry approaches to study protein glycosylation. Then tuning into the actual core of this presentation, glycoproteomics and glycoengineering. Throughout the presentation, and especially at the end of this presentation, I will give a case example of the work during my PhD, in which I have used the glycoproteomics tools to study protein O-mannosylation in humans, as Katarina just mentioned. We will then conclude, and I will leave space. I will be very happy to answer and interact with you through Katarina in any of the points that you might find relevant during this presentation. So when I approached the preparation of this webinar, I decided to structure it in a way that it can become a resource to whoever wants to approach the very fascinating field of glycobiology. So not only I will mention what our center and I specifically have expertise on, but I will also try to refer to what the field is doing. And I apologize in advance if I'm missing out on some key references or key points, but I hope this will serve as a beginning, as a resource for who's approaching the field. So let's get started with no further delays. And I would like to start with the concept of post-translational modifications. So perhaps most people that work in biology is aware that a protein, after being translated in a cell, can undergo a multitude of modifications. And the famous types of protein post-translational modifications can be, for example, phosphorylation, acetylation, and importantly for this presentation, protein glycosylation. So protein glycosylation, I should say, in a very biased way, is a very fascinating type of PTM. And one of the key aspects that I find really interesting is that protein glycosylation is present in all domains of life. And as a type of modification, it's a really, really diverse type of PTM. And this diversity is already visible at the level of what are the monosaccharides, the building blocks that make up glycans, so sugar that can be present on a cell. And if we look at what are the common human monosaccharides, we can see that more than 10 building blocks comprise and constitute make up the glycome of a single cell. So already at this level, you can see that there is a quite big diversity, that we can encounter quite big diversity in the glycome of a cell. In this presentation in glycobiology, you will be exposed to glycans with these colorful symbols for simplicity and also for a more understandable structure throughout the figures also of this presentation. These monosaccharides then are composed glycans, and glycans can, for example, be linear or branched, such as this glycan. They can have very different glycan composition. They can undergo modifications. For example, this glycan is phosphorylated. Glycans can be very small, such as my favorite glycan or mannose, or very big, such as this long GAG chain. Another source of diversity is arising from how these glycans are conjugated to a protein, and seeing on different ways of linkages, we can distinguish N-glycosylation, C-mannosylation, and O-glycosylation. If respectively asparagine, tryptophan, serine, threonine, or tyrosines are modified with glycans. My expertise is mainly on O-glycosylation. And O-glycosylation itself is a quite diverse type of modification. And if we look at what are the protein glycosylation pathways that are found on a single human cell, and what are the types of O-glycosylation pathways, we can see that there is already quite a biodiversity already at the first monosaccharide that is conjugated to the protein backbone. In my PhD that I have finished a month ago, actually, I have focused on O-mannosylation, where a mannose sugar is conjugated directly to a side chain of serine and threonines on proteins. So you will, throughout the presentation, you will hear a lot of examples of O-mannosylation represented with this green circle, or O-GalNAc glycosylation. It is one of the, perhaps the most characterized type of O-glycosylation in humans. So as you can see, this is a quite complex field. So whoever approaches this field might have the question, why do I need to study and why do I need to care about glycobiology? So to answer this question, I think it's crucial to keep in mind that if this modification is evolutionarily conserved, most likely means that it's functionally relevant. And this, in the past years, has proven to be true. Indeed, I will just show you these representative figures to show what are the multiple functions that glycans can have in cell. For example, glycans have been proved to be important in cell-cell adhesion, in binding to ligands, in receptor signaling, and many other functions. And this evidence is proved through, and I would say a seminal review and sentence in the field of glycobiology from Ajit Varki, one of the most important glycobiologists, where he already in the mid-90s, he suggested that all of the theories regarding glycan functions are correct. So this already gives you an idea of the importance of studying protein glycosylation. So not only glycans are functionally and biologically relevant, but also I should say that the relevance expands to human health. And to give you an example of how glycans are important in human health, I would like to mention a class of congenital disorders, which is called congenital disorders of glycosylation, that arises from malfunctions and the mutations, essentially, in the genes that encode for glycosyltransferases and other proteins involved in protein glycosylation. And just by looking at the numbers of disorders that are connected to glycosylation discovered throughout the years, we can see that there is an exponential growth, and more these congenital disorders of glycosylation are being discovered. So I will not talk more about this until the end of this presentation, but I will refer you, if you're more interested in this, in more general terms in glycobiology, I will refer you to some of these references. And in particular, I will refer you to the book, Essentials of Glycobiology, which is really an important book for whoever approaches the field of glycobiology that I can only recommend. And throughout the presentation, you will see these gray boxes here that, for example, report some references and some key take-home messages that you might find important, and important that you can come back to by looking at the recordings. So, as you can see, glycobiology is relevant, but studying glycobiology comes also with several challenges, especially in the analytics. And the challenges mainly are due to the complexity of this type of post-translational modification. The first source of complexity comes from the fact that glycans are synthesized in a non-templated way. And this means, for example, just to give an example, it's completely different than what happens, for example, for DNA replication, transcription, and translation, where a template is present and the machinery kind of follows this template. For glycans, this is not exactly the same. Instead, more than 700 enzymes in a mammalian cell contribute to the formation of the glycome, among which around 200 glycosyltransferases are responsible for protein glycosylation. And this quite small picture just gives you an idea of how many pathways, how many enzymes can be involved in this process. I would suggest you, if you are interested more in this, to check out this reference here. That is actually from our center that describes to 2020 what are the pathways mainly in humans that drive glycosylation. So among these, another source of complexity, analytical complexity, comes from the fact that despite being a common PTM, it's relatively scarce in a cell. If we compare it to the number of non-glycosylated peptides, glycosylated peptides are in quite a strong minority. In addition, generally, except for certain classes of very few classes of glycans, there is a lack of consensus sequences. So we cannot directly predict from the amino acid sequence where a glycan will be found. And in addition, glycosylation is often classified as a labile modification, and this lability comes from some analytical challenges that we will discuss more later in the next slides. So all in all, this complexity, I think from this complexity, we can understand that there is no single method that can answer all questions in glycobiology. Instead, several methods, several techniques have been developed in the past years for the characterization and investigation of the glycome of a cell and its functions. Among these, I think it's quite safe to say that mass spectrometry is a key technique in characterizing glycoconjugates and in more specifically glycoproteins. And when it comes to mass spectrometry, which is the focus also of this presentation, there are several approaches to this. And a traditional approach, which I informally call glycan-centric, has been characterized a couple of decades ago, at least, where glycans are removed from the protein backbone, or they are released in glycobiology terms, from the protein backbone to reduce sample complexity and be able to study them independently from the protein backbone. And this can be facilitated, for example, among the rest, with the use of glycosidases, so endoglycosidases or exoglycosidases. Among probably the most famous is the PNGSF, or endoglycosidase F, that aids the release of N-glycans. So these glycosidases release glycans, which can be analyzed with glycomics approaches, for example, using MALDI-TOF and SMS. So glycan-centric approaches are not the focus of this presentation, but I would like to just introduce you some pros, cons, and sample types that can be used for glycomics approaches, as well as some key references that you might find interesting in approaching this technique. I should say that Katarina, our moderator of today's webinar, has a lot of expertise in glycomics approaches. So should there be some questions regarding glycomics, I think Katarina is much more qualified than me in this. But in general, so I will leave here these boxes as a reference for, if you want to come back to this presentation as a recording. But generally speaking, glycomics approaches are really robust approaches that give great information on the structures and the linkages of the glycans. Although they come with the limitation that there is no information on the protein and the site from which the glycans are coming from. For these other mass spectrometry approaches, more protein-centric approaches have been developed in order to try to understand glycans in the context of the protein backbone. And to make a rough classification, two major types of protein-centric mass spectrometry approaches are top-down approaches and bottom-up approaches. Some of you might be familiar with this, but we'll just go through quickly for the first to go then into bottom-up approaches later. So top-down approaches see the direct analysis of intact proteins, so without digestion into peptides, for who has experience with mass spectrometry. And the typical outcome from this analysis is a spectrum like this, where we can see how many glycans decorate a specific protein that we use in the analysis. Also in this case, some pros, some cons, but the major pro is that we have information on the heterogeneity and the site occupancy of different glycans on a protein. For example, we can see in this protein, there are 61 GalNAcs modifying the vast majority of the proteins. Although these top-down approaches come with the limitation that they are not applicable to complex mixtures. Instead, Bottom-up approaches are more suitable for this. Bottom-up approaches consist in the analysis of peptide mixtures, so proteins that are that essentially are broken down into smaller peptides, also from complex samples. And bottom-up approaches are key, in my opinion, to study protein glycosylation at a site-specific level, but on a proteome-wide scale. So we can have information on more proteins at the same time, still keeping a site-specific resolution. And just to give you an idea of the outcome that we can expect from a bottom-up glycoprotein analysis is, for example, that we can see that we can map exactly what glycans are present on which specific amino acids on a specific protein. In this case, we have mannoses and galax modify a mucil-like domain of a protein. So the bottom-up glycoproteomics analysis is the focus of this presentation. And as you can imagine, there are just like many techniques in biology, there are a lot of ways of doing bottom-up glycoproteomics analysis. Although a general workflow for glycoproteomics looks like this. So if we start from an input sample, for example, cells or tissues, what we will do first is to extract proteins, and then protein extract undergoes digestion into peptides, followed up by an important step that is enrichment of glycopeptides before mass spectrometry analysis. So I will come back to all of these steps in the next slides, and also some other steps that can facilitate this process with some specific questions. So let's start with the input material. So when it comes to the input, in virtually any sample, you can extract proteins from, which can be cell lines, tissues, organs, and even small organisms, can be used as an input for glycoproteomics. So here, I would like to give you just a rough rule of thumb on the quantities. So if we talk about a sample with a low complexity, for example, a pure protein or a mixture of very small amount of protein or very little proteins, a few micrograms are enough to get a quite good glycoproteomics analysis. When it comes though to more complex analysis, for example, from cell lysates, here I think milligrams are required to have a good analysis, a good glycoproteomics analysis at a site-specific level. As a rule of thumb, as an indication, when it comes to cell lines that I have more experience with, a rule of thumb is to start in volume from at least 150 microliters of packed cell pellet, but this is of course just an estimation. If we have room for decision of which sample and which cell line, cell model to use, it's important to select wisely what model to use. I will leave you here some references that discuss about some of the factors that should be kept in consideration if you need to choose a model to study glycosylation. And in particular, the first reference is a big recommendation of mine because it really introduces glycoproteomics and the workflow of glycoproteomics in a very detailed way. And from this reference, I have also taken inspiration for this presentation. From the same reference, we find also this table, for example, that gives an idea on what are the possibilities from different models in terms of, for example, labeling and other things that we will see in the next slides. So other things that I would like to highlight when it comes to selection of the cell model is to evaluate the expression of the glycosyl transferases. So is the glycosyl transferase required for the glycosylation I want to study expressed in this cell model? And next, is it possible to simplify the glycome of this model? And I think this point is really important for the sake of this presentation because it will bring us to one of the core techniques of these slides. So what do I mean with simplification of the glycome? So the rationale of simplifying the glycome comes from a need to improve the analytics. Indeed, if you remember, glycans are labile, and especially large glycans are really ionized really poorly in mass spectrometry. And having a simplified glycom simplifies also the data analysis and the interpretation from our spectrometry. It also increases the homogeneity of a sample, so the information that we can get out of glycoproteomics analysis, and also in a more technical way, it simplifies the glycopeptide enrichment step, and this will come later in the slides. Regarding the simplification approaches, I will leave you here some key references. And from the first reference from our center, I have taken this slide that summarizes the three major types of engineering that we can do to simplify the glycom of a cell. In brief, enzymatic engineering, metabolic engineering, and genetic engineering tools are used in glycobiology to simplify the very complex glycom of a cell line. I will not talk much about enzymatic and metabolic engineering tools, but I will rather spend more time on the genetic tools, which lead us to the glycoengineering part of this presentation. So why would we use genetic engineering tools, and why have they been used for the characterization of the glycoproteome of mammalian and eukaryotic cells, among the others? So the rationale of using a genetic approach is very simple, and I will explain it with some numbers. Starting from 3 monosaccharides, the ones that we saw in a few slides ago, the combinations that we can do using different linkages, different combinations of glycans go up to more than 1,000 unique trimers. And as you can imagine, approaching the glycoproteome from a glycom point of view is a very challenging approach, because you can imagine how vast the glycom can be, at least in theoretical lines. A more feasible entry point is instead the glyco-genome, where about 250 enzymes are responsible for glycosylation, as we saw some slides ago. And this entry point has been used also by our center in the generation, among the rest, of the simple cell technology. So the simple cell technology developed about a bit more than 10 years ago now, aimed at reducing the complexity of the glycom by preventing the elongation of the glycans. So, and this has been achieved by knocking out key genes encoding for elongating enzymes. So what we will see from a simplification of specific glycans is that we will have a more feasible characterization via mass spectrometry, a more feasible data analysis, and a higher throughput. And so the simple cell technology has been developed mainly for 2 types of glycosylation. This is the O-GalNAc type of glycosylation and O-mannosylation, where respectively a genetic knockout of the cosmic chaperone and POMGNT1 glycosyltransferase lead to a reduced complexity of O-GalNAc and O-man proteins, leading up to these respective proteins with a single GalNAc and mannose sugars. This allowed the mapping of the O-GalNAc and the O-man glycoproteome in human cells across different cell lines, and it has been a milestone in the characterization of what proteins can be glycosylated in humans. So in the next slide, I would like to introduce you more, like in a technical way, of how we do glycogenome editing. And so when it comes to glycogenome editing, we like doing knockouts in our center, inducing insertions and deletions in the genomic locus that change the frame and introduce stop codons in a cell line, for example. Very briefly, I will go through the procedure, but if you're interested more in these details, I will refer you to some references and also to this slide, in the recording. So the first step is generating guides. So the first thing that I would recommend doing is to design at least 3 or 4 guides that target the first coding exons of the gene of interest. And in this case, on-target and off-target scores are important in the selection. Next, these guides, what we do in our center is to clone these guides on a suitable backbone. And here we leave you with some gene numbers in case you wanna purchase and acquire these constructs. as well as some other all-in-one systems developed by other laboratories. After cloning these guides, we would validate them, validate the best guide by co-transfecting it in the target cells, extracting the genomic DNA and amplifying the locus that has editing, ideally the editing, and analyze the amplicon with a technique called indel detection by amplicon analysis, IDA. So unfortunately, I will not have time to go through this technique, but these three references are really key for the understanding of this technique. In any case, I should say that our center has deposited already a library, quite extensive library of guide RNA plasmids targeting most human glycogenes. So you actually might not even need to do these first three steps yourselves, but you can, scroll through AdGene, this page of our one of the professors at our center, and you might find a plasmid that could be interesting for you. So after acquiring the plasmid or cloning the plasmid, the steps are pretty linear. So co-transfect the cells with the guides plasmid and Cas9 plasmid, but enrich the transfected cells, for example, following GFP expression, let the cells recover, and then, apologies, and then go through a single cell sorting step to have isogenic populations of cell lines that then we can leave growing, typically a few weeks, and screen, for example, with IDA, as mentioned before, if possible, or with Sanger sequencing that is more accessible to most laboratories. Further validation is then desired, for example, by Western blot or immunofluorescence, if possible. So genome editing tools, as mentioned, they have not only been applied to the simple cell technology, but also to the dissection of glycosylation pathways, for example, in human cells. So typically what we will do is to generate a knockout of a glycogene, for example, on a simple cell background, that should abolish a specific type of glycosylation. Or then approach a discovery, for example, of new glycosylation pathways. We have also used this approach to investigate specificities of different pathways, or produce proteins, reporters, for example, with a homogeneous glycosylation pattern. More on these references here. I will not have the time to mention other glycoengineering approaches, but just quickly one thing that I would like to introduce you to is that it's possible to reintroduce or introduce specific glycogenes, for example, by genomic integration, antibiotic selection, or other techniques, also found in these references here. So as you can see here, in these slides, we have seen that the glycome can be simplified, and this can be a step that comes before the collection of your actual sample. So the next step after having your sample, the idea is to extract proteins, right? And depending on this is very sample dependent. But for example, if the sample comes from a complex from a tissue, the cells can be released from extracellular matrix, for example, with ETTA. The cells are then lysed. Here there are several methods. I will just leave them here for future reference. The lysates are typically then cleared by centrifugation. Proteins are reduced and alkylated to render them exposed to proteases, and optionally then cleaned up, for example, with C18. Protein extracts then undergo digestion into peptides, and also in this case, there are several ways to do digestion. Just for your reference, digestion can be done with the workhorse protease trypsin. That is a very robust solution for most applications. We also can also to evaluate non-specific proteases such as pronase, proteinase, or also O-glycoproteases such as operator or sticky. Also more on this on the references. But this is all depending on what type of sample you want to have. So and also other options are available as well. Peptides then can be analyzed via mass spectrometry, but there is one important step that I haven't mentioned so far, which is the enrichment of glycopeptides. So I believe that this is a really important step that is not trivial, and maybe who approaches glycobiology might not evaluate and keep in consideration from the start. So glycoenrichment consists in the removal of most non-glycosylated peptides from the sample that we are working with. And here is some literature about that. But I believe, as mentioned, that this is a necessary step for the analysis of complex mixtures. And this comes back to the fact that non-glycosylated peptides are in vast majority compared to glycosylated peptides. And simply because of these different amounts, we will not have an in-depth glycoproteomics outcome if we do a glycoenrichment step. There are several ways to do glycoenrichment, and mainly two different types, chemical and biological types of enrichment. For your reference, I would mention just some of them, but I will refer you to the very beautiful work from Nick Riley from Caroline Bertosi's group that has this really beautiful and comprehensive overview of the major types of glycoenrichment that can be evaluated. In my work in our center, we like using the first one, which relies on lectins for the enrichment. And lectins, especially used in the context of lectin affinity chromatography, are proteins themselves, mainly bacterial or plant proteins, that present a high specificity for glycans. And commonly used lectins that you might have encountered also in your work are, for example, CoNA, VVA, PNA, and others. Here is a non-comprehensive list of lectins. More lectins and also more applications of lectins can be found also in the essentials of biology. And this is the customary table with some pros and cons of lectin chromatography. And in brief, I think among these, it's I would say the most important pro of using lectins is that they can give you a glycan-specific information. So here, a specific lectin can enrich only a certain type of glycans, thereby separating, different hexose sugars and distinguishing mannose, for example, from glucose and so on. Although lectins present a high specificity for glycans, they have a quite weak affinity, and sometimes also broad specificities towards other glycans. So this is really lectin specific. So more on this on the references. However, I would like to give you a little bit of a practical workflow for lectin affinity chromatography. So what I have actually done in my work and what we do at our center. So still keeping O-mannosylation as an example, what we do is to use a long column, so a long and thin column that is packed with a lectin immobilized on a solid phase for our enrichment. So before doing an enrichment, as you can appreciate from this image, it could be an optional step that I would recommend, at least for most applications it's necessary, is to apply a glycosylation treatment that allows a more specific enrichment. And here I will give you just an example. So in my workflow, I use PNGSF to remove N-glycans before doing affinity chromatography with CoNA, because CoNA has affinity for essentially all mannose sugars, right? And more or less. And this would lead to an enrichment of mannosylated N-glycans, specifically if presented at the terminus of the glycan. So PNGSF might help achieving a more specific enrichment of lectins. So PNGSF is an example, but also sialidase treatments can allow a more deep characterization of the glycoproteome. So here are some references to this aim. And back to a more practical workflow, what I do in my center is to use, as mentioned, lectins conjugated to a solid phase, which can be saccharose or agarose. The good thing here is that a lot of lectins are already available, already conjugated to solid phases, so they are commercially available. If this is not the case, lectins can be either purchased or produced in-house and then conjugated, for example, with NHS chemistry. And the idea then is to pack the column. And this is really, something where creativity comes in, because this is a picture from my lab and one of the columns that I have used in my chromatography. The idea is that we need to force the slurry into a very small tube. And my trick is, for example, to use a syringe and HPLC connectors to literally force in the slurry in this small tube. And the use of a small tube actually comes with some advantages, and it kind of mitigates the weak affinity of lectins just by having more lectin resin that your sample can interact with. So this is a little bit our trick in Copenhagen. And the idea is that this column can be equilibrated and run on an ectopurifier typically. Just for reference, we use 100 microliters per minute at 4 degrees. And the buffer is really lectin specific, so this needs a little bit of research on how the lectin works. But just to give you an idea, for the sake of open science, this is more or less how chromatography looks like. So you would inject your sample. A peak comes out. These are the non-glycosylated peptides, so the ones that do not bind to your lectin. Typically, what I like doing is to leave the column equilibrating before injecting or switching to an elution buffer that allows the competitive release of glycosylated peptides, which can be collected in different fractions, allowing also a fractionation that improves even more the depth of the glycoproteomics outcome. So this is a little bit in a nutshell what we do here in Copenhagen. And this is the workflow so far. So we have seen, there are some optional steps. Last thing that is remaining is the mass spectrometry. So before running mass spectrometry, it's important to do a CT purification, stage dipping, to avoid contaminations on the mass spectrometer before going into the bottom-up mass spectrometry approach. So in the interest of time, I will go also quite quickly on the mass spec part. But I think this slide summarizes the main points on a typical workflow for mass spectrometry. So at least what we do in our center. So what we start with is to separate the peptides with a reverse phase seating nano-LC chromatography on a nanoflow, which separates the peptides based on hydrophobicity. Peptides then undergo electrospray ionization. This can also vary, but this is our setup at least. And the mass spectrometer then, typically our Orbitrap mass spectrometer. In our center, we have Orbitrap Fusion or Orbitrap Fusion Lumos instruments. The first scan essentially looks at separates the intact ions based on the mass to charge ratio, to allow selection for fragmentation. And the MS2 fragmentation is really important because it gives us the information on the site and the sequence of the peptide. And typically in our center we do fragmentation, HCD fragmentation. So this is a little bit technical, but in synthesis it allows sequencing of the aminoacidic part. And we combine it also with the ETD fragmentation, where which is a more mild type of fragmentation that leaves the glycans more or less intact and conjugated to the protein backbone. So it's possible to evaluate also hybrid methods, for example, ETCAD or ETHCD, which allowed the characterization and the discovery of the glycoproteome in, for example, in human cells. So more on these can be found, for example, in this reference, and the references in a slide, in a couple of slides. And so the outcome from this analysis, of course, needs to be analyzed. And this is a quite tricky part that I will also go through quite quickly. So the data analysis, in the old times, but also still now, manual inspection. And this typically, consists in looking at the spectra and trying to map what each of the peaks, is coming from. So which glycan is coming off and so on. This, as you can imagine, is quite expert eyes. It's more accessible and more. It's much easier to use softwares, for example, Protein Discover, FragPipe, or Bionic, for example, that treat glycans as variable modifications. So just quickly, this is a list of some search enzymes that can be evaluated. The problem is that these search enzymes are mainly designed for proteomics. So glycomics also, as Katarina mentioned, is quite of a growing field, and it's There is a lot to do still also in terms of analysis, although some glycoproteomics specific tools are emerging more and more in these years. I will leave you here also with some alternatives for who has a little bit more experience in mass spectrometry. But in practice, what we do in our center is to use a Proteome Discoverer, which is very user-friendly, though it's you need to pay to have access to Proteome Discoverer. We also evaluated MS-Fragger, which I would really recommend for open search, so unbiased glycan identification. So if you don't know what glycans you have in your sample, this could be a tool that you could look into. So yeah, so this is a little bit about mass spectrometry. This is the usual table with some pros and some cons. Main pro is the high throughput approach. Main con is the shotgun approach. So you will never have a complete coverage of the proteome. So this needs a little bit of attention. But I will leave you some references. I hope these are probably not comprehensive. I know that many other groups are doing great glycoproteomics, but I think this could be a starting point for further research. And yeah, so last thing that I would like to mention on a more technical level is one of the pros of doing bottom-up glycoproteomics is the possibility of doing quantification. So what is quantification? Quantification is the possibility of comparing, in our case, glycoproteomes of 2 or more different samples. So just to give you an example, you might would like to compare the glycoproteome of a wild-type cell compared to a knockout cell, or control cell line against a treated cell line. Or you might have a time course that you wanna, that you would like to analyze in terms of glycoproteomes. For example, answering the question if there are differences in the glycoproteome across different treatments or in time. So for this, it's isobaric labeling is very useful, and labeling can be done on protein level, SILAC, just mentioning, or on peptide level, which I have more experience with for example, dimethyl labeling, diethyl labeling, or TMT labeling. Also here, some key references for this type of labeling. And the first review can also give you an idea, a more general picture of what's happening. And in the last minutes, I would like to show you, how I have applied quantitative glycoproteomics in my work. So, for example, a typical approach that I have done in my doctoral studies was to start from simple cells that now we know that they have a simplified glycome, on which I have applied a knockout of a specific glycogene that I wanted to look into. Now, these 2 cell lines became the control and the case cell line. So these 2 cell lines were then treated in parallel. So I did a total cell lysate and digested proteins into peptides. These proteins these peptides were then labeled with 2 different labels. For example, heavy and light. In this case, during my PhD, I used a lot diethyl labeling. So essentially, these 2 populations can be mixed 1-to-1, and now you have 1 sample for glycoenrichment. So what I did was to do glycoenrichment with CONA and quantitative mass spectrometry analysis. So what I would have as an output for glycoproteomics workflow of this type is that I would see that some peptides lose glycosylation upon gene knockout, and this is visible as a ratio between control and case peptides. And then, another possibility is that I could reintroduce the glycogene or a mutation or a mutated glycogene to this cell line, and I would see that I would detect a complete rescue of glycosylation with the knocking of this gene. So this is just to give you an idea of what type of work can be done. And this brings me to the overview of the glycoproteomics workflow as we do it in the Copenhagen Center for Glycomics. So I will leave you this slide here for reference, as a kind of a comprehensive view of what we have discussed today and what I haven't discussed but can be done as well. So I would like to very quickly show you a last example of how I applied this workflow and how it's relevant for my research, how it was relevant for my research. So if you remember, I have worked with O-mannosylation And one of the research questions that I had was to understand the biosynthesis of O-mannosylation. And what was known before the beginning of my PhD was that there was an unknown pathway for O-mannosylation that was still to be discovered. So here I'm skipping a little bit of details, but you will find, if you're interested, there are some references in this slide. But in order to try to understand what was this O-man enzyme, I have used glycoproteomics approach. So we had a candidate protein, Tm260, that was also mentioned by Katarina in the introduction. And Tm260 struck our attention because it showed structural similarities with other O-man enzymes already known. For example, some yeast homologs, but also other human O-man enzymes. So how I approached this with a glycoproteomics approach and glycoengineering approach was to use HEC293 cells, which I established a knockout of Tm260. And I applied these two cell lines with a differential glycoproteomics approach. So what I saw from these analysis, with the workflow that you saw in the previous slides, was that a Tm260 knockout led to a loss of non-glycosylation on specific peptides, the ones that we were interested in. Suggesting that Tm260 that Tm260 is important for this function. I then did a confirmation by knocking in again Tm260, which correctly expressed with this Western blot and completely rescued the glycosylation effect that we saw. And finally, I knocked in a catalytically inactive version of Tm260, which expressed correctly, but was completely unable to perform glycosylation, suggesting that Tm260 was a protein-specific O-man glycosylation transferase that constituted a novel O-man glycosylation pathway. So as a last point for this presentation, I would like to give you the relevance of this discovery and of glycoproteomics approaches. Indeed, not much was known on Tm260 at the time of this discovery, other than the presence of biallelic variants of Tm260 gene that cause a rare disease called SHDRA syndrome, presenting with cardiac, renal, and central nervous system disorders in young patients. So in this work that you can find in this reference, in the first reference here, we have assessed two published variants, and reported two new variants from two new families with variants on the gene of Tm260. So the approach here was to generate cell models like we have seen before in the presentation, in which I knocked in Tm260 knockout cell lines. I knocked in variants genes of Tm260, which I could assess with differential glycoproteomics approaches. And what I could see that the four variants that I assessed they had a reduced ability to rescue O-man glycosylation on these specific domains that Tm260 targets. So suggesting that SHDRA syndrome is a new congenital disorder of glycosylation. Therefore, giving you an idea of how glycoproteomics can shed light also on human disease mechanisms. With this, I would like to conclude you. I hope this presentation was useful and that it could give you some insights on how we can approach glycosylation with a glycoproteomics work. All of this work and the things that I have presented today wouldn't have been possible with people at the Copenhagen Center for Glycomics. And here I just mentioned some of them that were directly involved in this work, in my experience, and in particular the people in my group and the collaborators. However, I would like to thank everyone that was involved in this, also all the people that were very inspiring and gave me also tips on how to build this presentation. I would like to thank all of you, importantly, for tuning in today, and I will be very happy in the time remaining to answer questions and to give you a little bit more insights on these topics. Thank you very much.
Thank you, Lorenzo. That was really, really nice, comprehensive overview of the methodology with an application in the end that actually is showing that it can end up with a discovery of a new O-mannosylation pathway. So thank you so much. It was great. I will read some of the questions, and in the meantime, I encourage all of you to write your questions in the chat or Q&A. Let's start with, there's a question on, can you use the variation of this approach to look at glycosylation patterns in, for example, healthy and diseased cell lines?
Absolutely, absolutely. That's a very important point. It's a very important point. And I think for this, the quantitative approach that I mentioned towards the end of the methods part can be useful for this, because we can actually compare a healthy cell line with a disease cell line, if we can use these labels. Yeah, so absolutely this can be done, and it can also give really important information, so translational information. Absolutely.
Okay, thank you. And we have another question. What, in your opinion, controls what glycoforms are expressed over the many possibilities of combinations and diversity that exist? For example, on N-glycosylation.
That's absolutely that's a absolutely good question. So this is indeed object of study. It's it's absolutely a good point, because it's true that virtually there are so many, I mentioned those thousands of combinations that can be formed with 3 single monosaccharides. But in reality, what we have seen is that only a certain limited amount of structures and glycans can be found in a cell. It's still a quite large number, but there are some ways in which cells can select what to do. So this all boils down to how the glycosyltransferases work. What are the specificities of the glycosyltransferases? So what specific proteins, for example, can interact with the glycosyltransferase, can determine what structures can be presented. Additionally, also the location of the glycosyltransferases, for example, in the secretory pathway, can drive and can make some changes, right? Some glycosyltransferases are present in the ER, in the endoplasmic reticulum. Some others are in the Golgi, the early Golgi, in the late Golgi. So there is also some there are some differences also in that sense. So there is no simple answer to this, but it's an important point. It's an important point. This, I don't know if you would like to add anything, Katarina. But
I think that was perfect.
Yeah, it's something
Thank
really
you.
important, though.
But I would like to ask you one question. I mean, this you gave a very, very detailed overview of the methodology, but I was just wondering, what are the critical points in this protocol? So what are the points where everything can go wrong if I don't do it well?
Yeah, okay. I see. That's a very good question. So if I need to pick, I think a very important point is the enrichment step. So I try to underline that part, because it's actually quite crucial. It's a quite crucial step in the protocol. A good enrichment can give you a really good glycoproteome analysis. A bad enrichment can also lead you to very misleading results. So, for example, you might not see important peptides and lose important information if, for example, you use the wrong enrichment method. So I think that's the key step that is important. Of course, other steps are important as well. So a good trypsinization, a good trypsin digestion is required to have peptides and not intact proteins. Although the other steps are relatively more robust. Another important step, I think, is how you would search your mass spectrometry data. So here there is a whole, we could do a whole new webinar on this. I'm not the right person to do that, but I think also looking at the large amount of data that you can get from mass spectrometry in the correct way is crucial to have the information that that you need and that can be relevant, biologically relevant.
Okay, so and if you take out the sorry. Continue.
No, I would say these 2 points are, I think, the most crucial.
Okay, yeah. So if you take out the glycome simplification step or glycogene editing, how long does it take to do this protocol in the lab? From start of the cell tissue step until mass spectrometry and data analysis?
Well, per se, it's like if we exclude the genetic engineering part, which can take, I would say, maybe 3 months in average, let's say, in our hands, the actual glycoproteomics sample prep is not that long. I think in a week of daily work, you would get a sample from mass spectrometry analysis. What comes, and then afterwards there is the analysis part. The run per se is relatively quick, but I think it's around a week, I would say.
Okay.
Because there are
Perfect.
some overnight incubations
Sounds
here and there.
feasible.
So a week.
Sounds feasible. Okay, we have another question that you partly covered already, but so there's a question: can one amino acid within a protein be glycosylated with different sugars, resulting in different downstream signaling?
That's an important question. So in the interest of time, I haven't introduced an important concept in glycobiology, that is the heterogeneity. And I think this is something that can be found also in the essentials of glycobiology. But what is important to know is that this is possible. Like a single site, there can be competition for a single site for different types of glycosylation. And this has been also reported also in our field, like in the field that I worked with the O-mannosylation and the organic glycosylation. It can happen that the same site can be targeted by different glycosylation pathways. So this is absolutely possible, and it's something to absolutely keep in mind. And I will add also another thing. It's well understood that also other types of post-translational modifications can compete and can coexist somehow with the protein glycosylation. For example, competition between O-glycosylation, especially nucleocytoplasmic O-glycosylation, is in strong competition with protein phosphorylation. So there is a whole very beautiful literature on this as well. So absolutely, this is something that can occur as well.
Okay, thank you. We have some new questions in the Q&A. So Muhammad is asking, what method do you think is suitable to remove N-glycans on CD47 protein? Do you think that glycosylation affects or influences surface expression of membrane surface protein and thereby interfering with immunotherapy?
Okay, so thank you for your question. So for your two questions. So for the first part, so I do not have direct experience with this protein. So I cannot specifically say what enzyme can be used for this. But a workhorse enzyme for release and the removal of N-glycans from a protein that I would recommend starting with is PTMs, because generally it works on like most N-glycan structures. Other endoglycosidases can be used. For example, ENDO-H can be used, and it can have a partially non-overlapping activity with PNGSF. But these are some of them, and I would recommend also going back to the references on the glycosidases for this. But in theory, unless I'm missing something, some biology in this specific protein, in theory it should be possible. For the second part, if I remember the question correctly, is if glycosylation interferes with the presentation of the protein to the surface. Yes. So also in this case, I don't have information specifically for this protein, but in literature, it's been reported that glycosylation is important with the cell surface presentation of specific receptors and or secretion of proteins. So it could very well be that the N-glycan you're looking at, or more glycosylation events that you're looking at, they're important for the secretion and the presentation of this protein at the surface. So absolutely, this is a possibility. And together with this, literature also shows that the maturation of certain receptors can be facilitated, if not even enabled, by specific glycosylation events. So absolutely, this is something that could occur also to your protein of interest.
Thank you, Lorenzo. We have some more questions. What is the best approach to deconvolute glycan data, and which platform is best to start with unknown glycan composition?
With a known or
unknown
unknown?
Glycan composition.
Yeah,
I'm tempted to say a calculator. That's what I use.
I think actually, Katarina, you might be actually more qualified to answer this question, given your glycomics background. But this is a question that is, you know, I don't have to, I must say that I do not have much experience in this. But so other than with this software that I mentioned, this MS Fragger software, I will go back to the slide if I can find it quickly. That could help in the unbiased. Here we go. In the unbiased identification of what glycans are there. So I like MS Fragger. It's a quite user-friendly software. It's becoming more and more user-friendly and more versatile. So especially like in my case, I have used it in the FragPipe system. And here is actually the reference for MS Fragger, one of the references. So I would perhaps start with this. Like in my experience, I have a very good experience with this. Although I must say there are several other options in literature that you could explore and might be more suitable for your case. Of course, what Katarina suggests is to use the calculator, so do an approach like this one, so trying to understand, you know.
That was all, I mean, partly a joke, but I often use only the calculator. But there are also tools like XPaC, Glycomode,
exactly.
or even GlycoWorkbench that help with the analysis of unknown glycan compositions.
Absolutely. Yeah, absolutely. So yeah, that's
then
what I would suggest.
Okay, shall we move on to the next questions? We got a few more. Can you discuss any recent advancements in glycoengineering that have translated into clinical trials or therapeutic applications?
That's a very good question. So here on top of my mind, I cannot give you a very quick example, I must say. I don't want to, give you wrong answers on this. But I will maybe come back to you. If you post the answer on the question and answer session, maybe I will give you a little bit more informed answer on this. Perhaps I'm missing something, you know, evident. I don't know, Katarina, if you are.
Yeah, I cannot get anything out of my mind right now. Maybe we can go back to that later.
So for therapeutics.
Yeah. I mean, we already know so much about how glycosylation influences the function of antibodies, right? And these are commonly therapeutics. So
exactly. If we consider,
this could be an example. And this is where we know mostly about the function of glycosylation in this particular proteins, right? So how glycosylation influences IgG or immunoglobulins general.
That is absolutely true. Yeah, that's a very good example. That's a very good example. Yeah.
Now we have another question. How many cells should I start with to perform this protocol that you show here in the overview? Do you have a recommended protein extraction protocol to enrich for secretory pathway proteins and avoid cytosolic proteins?
Okay. I will go back to, let me talk about if I can find the slide here. I think this slide could do. So your question is so you are essentially interested in proteins in the secretory pathway. That is actually quite of it's an important question, and actually I faced with the same challenge during my doctoral work actually, and I have recently reported on a bioarchive paper strategy that we explored in our laboratory for enrichment of transmembrane proteins and proteins in for example, in the secretory pathway. And this involved the use of digitonin for permeabilization of the cells and release of soluble cytoplasmic proteins that I was, for example, in my case, not really interested in. And in that way, I could solubilize the proteins in the secretory pathway and in the organelles, and importantly, in the membranes, using a mild detergent, for example, MP40, while keeping the nuclei intact. So I can, if you are interested, you can check out, let me see if I can find my own, yeah, I don't want to reference myself, but perhaps you could, you could find some inspiration in what we have done there. Let me see if I can find my own work. Yeah, this one. So you could find some inspiration there. Of course, there are several other ways to do it, but this is one thing that we have explored recently. But I think already doing a nuclear prep already eliminates quite a lot of proteins, nuclear proteins that you might not be interested in. And so, yeah, this is one approach that you could explore, for example. I hope I have answered the question.
And this, there's an additional part from the same question. For someone who does not know how to interpret mass spec data, how might the data be presented?
Also quite a relevant question. Indeed, it's often really challenging to to compact information into into something usable, right? So this really, I would say, there is no simple answer to this, because it really depends on what is the main focus of the work, right? So if you're more interested in reporting, for example, what proteins are glycosylated, then you could, for example, show lists of proteins, or the volcano plots if you're doing replicates and quantification. Or if you're interested in specific sites of specific proteins, you could show the old good sequence, which is not really appealing, but it could give you some like a specific and immediate information on what you want to convey. So for this, it's really there is no simple answer to this, but I would recommend you getting some inspiration from other papers, from what other people did. And importantly, another thing that I would like to suggest that also helped me is to look at papers for other post-translational modifications. For example, phosphorylation. It's the concept is very similar. Of course, we are working with a very different modification, but the type of data is quite similar. So perhaps you could get also some inspiration from phosphoproteomics data on how to present data. So I hope this was useful.
Great, thank you. We even got a suggestion to look up the first in-human phase 1 study of a glyco-engineered monoclonal antibody in patients with metastatic and advanced HER2-positive solid tumors. So this is probably a good example of the previous question. Yeah. And one other question, it's very interesting, is something that we should always think about. How stable is the glycosylation? Should we take any precaution during sample preparation to preserve the glycans?
This is indeed an important point. So it's not something I have touched too much upon in this presentation, in the interest of time. And it's also not something that I have explored too much in my work, since I was quite lucky to work with glycosylation that is composed of a single mannose sugar. So, and importantly, I was working with mainly with the initiation of mannosylation. But there are methods for derivatization of glycans that can be used, and that's the term that you could look for protection of especially terminal glycans. And my thought goes to sialic acids. Those are the first ones that go, and that are very difficult to characterize and analyze. So there are absolutely methods to stabilize, especially terminal sugars. So I think for this, we could, I think also the essentials of glycobiology could be a start. I don't know if you have more specific suggestions, Katarina, from your...
Perhaps the linkage specific ethyl esterification, basically you could derivatize sialic acids in a way that you can distinguish which linkage, how are they linked to the galactose. So in this way, not only that you preserve the sialic acid, for example, during mass spec MALDI analysis, MALDI-TOF analysis, but you also can get a little bit more information on the actual linkage, and which is actually very important, since it's been shown that, for example, 2,6 linked sialic acid is differently expressed in cancer. So this is something that has been published maybe 5 years ago, and it could be a method to preserve very labeled sialic acids.
Yeah. I also, that's a very good point. I don't have much experience on this, but I know, for example, permethylation is one of the, there are chemical ways, to preserve, especially sialic acids, but generally there are also some other, it's mainly chemical strategies for that. So, yeah. But good point, absolutely good point.
Yes. If you have any more questions, put them in quickly. I see many very nice comments. Thank you for your presentation and fantastic talk.
Thank you for joining.
I don't think we have any more questions.
Yeah, but
I hope I have not missed any.
Yeah.
Oh.
If you have other curiosities, other, anything you would like to talk about, also, to interact, please. I know there is a question and answer session in the website that you can use, we'll keep an eye on it. And, or of course, you're very welcome to reach out. You can find our contacts in the Copenhagen Center for Glycomics website. So absolutely, please do not hesitate to reach out. And, if we can be of help, in some specific strategies, or if we can refer you to someone that, in our center or some other collaborators or people in the field can do, we would be more than happy.
Great. Thank you so much, Lorenzo.
Thank you. Thank you very much. Thank you very much, Katarina. Thank you, Renee. Really, it was a pleasure. And thank you everyone for joining.
Thank you both for being here and for a really interesting talk and a great Q&A session. And as I mentioned earlier, the webinar is being recorded and will be posted later, so you can revisit it. And thanks everyone for coming, and see you next time. Thanks.